Hello, my name is Jack DeCruz, and I'm a seventh grader at Menlo Park Academy. My topic was, what is the best product to clean your hands? I thought of this when a bottle of antibacterial liquid soap was replaced with bar soap, and I wondered which removes more bacteria from your hands. Abstract. What is the best product to clean hands? This experiment tested various cleaning products against bacteria found on a kitchen sponge and a shoe sole. The hypothesis was that soft soap, an antibacterial liquid soap containing benzalkonium chloride, is the best cleaning product because soft soap kills bacteria and water washes away any remaining bacteria. In the experiment, a thoroughly cleaned hand washed with all the cleaning products was contaminated with a dirty kitchen sponge or a shoe sole. The hand was then cleaned with either Dove bar soap, Purell hand sanitizer, or soft soap, following CDC guidelines. It was then placed onto a petri dish containing nutrient agar and placed in an incubator. Every 12 hours, results were recorded and photos were taken. A hand contaminated with a dirty kitchen sponge was best cleaned by soft soap with 237 colonies, followed by Dove with 582 colonies, Purell with too numerous to count colonies, or TNTC, and the hand contaminated but not washed had TNTC colonies as well. In the case of the hand contaminated by the shoe sole, Purell performed the best with only 4 colonies, followed by soft soap with 92 colonies and Dove with 217 colonies. The hand contaminated but not washed had 324 colonies. The hypothesis that soft soap would clean best was supported with the sponge results that showed the least amount of bacterial growth. However, with the shoe soles, Purell was best, suggesting that the ethyl alcohol in Purell is better against bacteria on shoe soles than the sponge. Retesting the Purell would be desirable to check for consistency. Scientific question and hypothesis. Scientific question. What is the best product to clean hands? Hypothesis. If Dove, Purell, and soft soap each clean a hand contaminated by a dirty kitchen sponge or a shoe sole, soft soap and antibacterial soap will remove the most bacteria from the hand because it both kills bacteria and uses water to wash away remaining bacteria. Here's my material list. I had 10 petri dishes, antibacterial soft soap, Dove bar soap, antibacterial hand sanitizer, Purell, an incubator at 80 to 90 degrees Fahrenheit, tap water at 90 degrees Fahrenheit, gloves, dirty shoe, dirty kitchen sponge, and an opaque box that will hold 10 petri dishes and fit inside the incubator, and tape to make labels. My procedures included preparation. First, each of the 10 petri dishes will be labeled with white tape. The labels will be clean hand control, sponge no wash, sponge wash with Dove, sponge wash with Purell, sponge wash with soft soap, shoe no wash, shoe wash with Dove, shoe wash with Purell, and shoe wash with soft soap. The last one is the petri dish and agar control. To make your clean hand control, wash your hands with Dove and tap water at 90 degrees Fahrenheit for 40 seconds. Next, wash your hand with 1 pump of soft soap and tap water for 40 seconds. After a 1-minute air dry, apply 2 pumps of Purell hand sanitizer. Put your index and middle finger at the center of the petri dish labeled Clean Hand Control, and gently spread your fingers toward the outside of the dish for 3 seconds. For sponge no wash, you can wash your hands as the same method as the control. Next, hold a wet kitchen sponge in your hand for 5 seconds, perform a light squeeze to get some liquid out, and hold for 5 more seconds to contaminate. Lastly, place your index fingers and middle fingers on the petri dish labeled sponge no wash, and spread your fingers like before. For sponge wash with Dove, Purell, and soft soap, wash your hands the same way as in the control, contaminate with the sponge, and wash with either Dove, soft soap, or Purell for 20 seconds. Place your index and middle fingers into the corresponding petri dishes and spread your fingers toward the outside for 3 seconds. For the shoe soles, for Petri dishes 6 through 9, use the same process by cleaning your hands with the control. Next, instead of getting your hand dirty with the sponge, hold the sole of a wet shoe for 15 seconds. Follow the same procedure for washing the dirty hand with no soap, soft soap, and Purell. Put your index and middle fingers on the Petri dish the same way. For the agar and Petri dish control. For the 10th Petri dish, do not do anything and label it agar and Petri dish control. Incubation. Put all 10 Petri dishes in a dark box and put the dark box inside an incubator with a temperature of 80 to 90 degrees Fahrenheit. Post-experiment. After the experiment has ended, sterilize all of the Petri dishes by filling them with rubbing alcohol and put the dishes in a Ziploc bag and throw them into the trash. Hand contaminated with sponge raw data. So here's my data. You can see, as expected, the sponge that was contaminated but not washed, right from the beginning, had too numerous to count colonies. Next was Purell, which after 24 hours and onward had too numerous to count colonies. Dove was the next worst, and soft soap performed best. Clean hand control and agar control didn't go over 2 colonies at all. And you may notice as the time goes past, the amount of colonies decrease. This is because after the maximum number of colonies were achieved, the colonies on the Petri dish merged and could not be counted individually. This is the hand contaminated with the shoe sole raw data. It was worst cleaned with no soap, as expected. Next worst was Dove. After that was soft soap, and Purell performed very well and only had a maximum of 4 colonies. Here's my graph for the colonies from the sponge contamination. As you can see, the black line represents the sponge, the hand that was contaminated but not washed. It went up immediately after 12 hours, too numerous to count. The Purell was next, and after that you can see the Dove, and the best, the lowest, was soft soap. Here's the graph of the colonies from the shoe sole contamination. As you can see, as expected again, the hand that was not washed is at the top. Next is Dove. The blue line is soft soap, and right at the bottom, there is the Purell and the controls. How the hand cleaning products work. Dove bar soap. Dove bar soap has no active antibacterial agents. Dove works with tap water to wash the bacteria on your hands right down the sink. Purell antibacterial hand sanitizer. Purell hand sanitizer contains 65% ethyl alcohol, an active antibacterial agent. Ethyl alcohol is an amphiphil chemical compound, meaning it loves water and fats, both of which make up the prokaryotic bacterial cell's membrane. The ethyl alcohol bonds to the cell's protective membrane and is able to break it. When this happens, the organelles and genetic material spill out and are not able to function, and it quickly dies. The soft soap antibacterial liquid soap. The active ingredient in soft soap is benzalkonium chloride, an active antibacterial agent that attracts negatively charged bacterial cell membranes because it has a positive charge, and is able to break them open, letting all the organelles and genetic material out, therefore killing it. Types of bacteria found on the sponge and shoe sole that were grown on the nutrient agar Petri dish. I was curious to see what was growing on my Petri dishes, so to find out what types of bacteria I was growing, I took a bacterial smear to view under a microscope. To do this, I took a sterilized toothpick and took a small sample of some desired colonies and placed them on a drop of water on a microscope slide. Next, I fixed it by moving the slide over a candle lighter 3 times, and after letting it cool down and dry off, I dropped methylene blue stain. And after waiting a minute, I dipped it in distilled water to remove excess stain. As expected, I found both spherical and rod-shaped bacteria, Cocci and Bacilli. After doing research, I found some common bacteria on kitchen sponges, and the sole of shoes included Salmonella, E. coli, and Staphylococcus. These bacteria are prokaryotic single-celled organisms that multiply through binary fission and belong to kingdom Eubacteria. As expected, I also saw some mold on the agar plates. The mold I saw was a multicellular eukaryotic organism belonging to kingdom fungi. And so here are some images I took under the microscope. So on the left, there's a white colony that appeared on the agar plates from the sponge. And on the right, there is a yellow mold colony that was on the petri dishes from the sponge. Here are the shoe sole microscope bacteria images. So on the left, I have an orange colony that I found on the shoe sole. And on the right, there's a white that was a white colony that was found. And here are the pictures I took and how I gathered my data. So these are the sponge petri dish images after 72 hours. So there's all the way on the left, the hand washed with Dove, then the hand washed with soft soap, hand washed with Purell, and the hand that was not washed. Here are my shoe sole petri dish images after 72 hours. All the way on the left, there is hand washed with Dove, hand washed with soft soap, hand washed with Purell, and hand that was not washed. Conclusion. The goal of this experiment was to find the best type of cleaning product for eliminating bacteria on your hand. The hypothesis was that the antibacterial liquid soap, soft soap, would remove the most amount of bacteria from a contaminated hand because it both kills bacteria and uses water to wash away remaining bacteria. The hypothesis that antibacterial soft soap would eliminate the most bacteria was supported with the sponge because it had the least amount of bacterial growth. With the sole of the shoe, the hypothesis was not supported because the Purell showed the least amount of bacteria. This may suggest that the ethyl alcohol in Purell is more effective against bacteria found on the sole of shoes. This experiment was successful because the bacterial levels could be observed and the bacterial levels varied in a predictable manner. Additional testing on this subject could include retesting the Purell to check for consistency. Thank you for listening. I hope you learned something.